Why haven't we gotten better Alzheimer's drugs, in your opinion? I heard a statistic like there's been 400 failed clinical trials. Most of the people who've gotten these drugs have some significant cognitive deficit before they go on the drugs.
Is most commonly associated with Alzheimer's disease.
The formation of these were causing neuron death and Alzheimer's. You hear people talk about Alzheimer's as type 3 diabetes. There's still a lack of clarity around what Alzheimer's disease is at the molecular level. What can somebody today in their 30s or 40s do to really meaningfully reduce their risk of developing Alzheimer's disease? We know can have a huge impact. One of the probably most potent ways to broadly maintain health, including brain health.
I do think is just crucial.
I've worked in dogs with the Dog Aging Project. They don't get exactly Alzheimer's disease, but they get dementia. The statistics are maybe 15% of dogs will get this during normal aging. Do you think we will get to the point where we have a cure for Alzheimer's disease?
20 years from now, I really believe that we will have solved the problem of Alzheimer's.
So, Cory, thanks so much for coming down to Optispan headquarters today. I'm really looking forward to this conversation. We'll do sort of a deep dive into Alzheimer's disease, talk some about systems biology and digital twins. But before we get into that, I'd really like to just hear from you sort of how you got started in this space and, what was your path to ending up at ISB and doing the work that you're doing now?
Yeah, so I did my PhD actually in breast cancer, looking at the estrogen receptor. And trying to understand really the transcriptional profile that happens when you put estrogen on cell lines and how that pertains to cancer and growth and stuff. And I very much enjoyed that. I did my PhD at University of Illinois in Urbana-Champaign. But there was also something to me that I wanted to understand more than just one pathway that was going on in a cell. And so systems of biology really sort of was attractive to me. And at that point, Nathan Price was there. He had just started as a new professor there. And so I joined his group as a postdoc with the idea of, okay, I'm going to, instead of looking at estrogen receptor, I'm going to look at the EGFR, and again, try and understand this in more of a sort of a systems approach. And at that time, we were working on glioblastoma, so in the brain, but not yet neurodegeneration. And so from there, then Nathan moved back to Seattle. I came with him here to Seattle, joined Institute for Systems Biology, and that was about 13 years ago.
Right. And where was Nathan at before he came to ISB?
He was at University of Illinois in Urbana-Champaign. Yeah,
okay.
So he was right there. So,
got it.
Yeah, where I had done my PhD and then joined him as a postdoc. And so then moved here to Seattle to do work with Lee Hood and Institute for Systems Biology. And then we actually I was already working in the brain doing glioblastoma, and we got a project that was actually the first project that was called the Accelerating Medicines Partnership in Alzheimer's Disease. So this was a big consortium that the NIH funded. And the goal of the consortium was to generate a lot of multi-omic data on the brain. And so that's when I first moved onto Alzheimer's as a project and helped to generate RNA-seq data on hundreds of postmortem brain samples. And so from then on, which was again like 12 years ago, that's when I started in Alzheimer's research.
Cool. And definitely we'll get into that. Just sort of set some frameworks here. I think you're at the Institute for Systems Biology. You said that phrase already a couple of times. I think a lot of people maybe don't know what systems biology even really means. So maybe you could give kind of your definition of systems biology and how it is different from other types of investigation that
sure, yeah,
that people do.
So it's funny, even internally we have lots of discussions of what is systems biology, and I think you ask you're going to get a different answer from anybody you ask. But for me, one of the things that I like, or the way I define systems biology, is that we know there's a sort of reductionist approach to science, right? Isolate the variable, understand the variable's contribution to a system. Systems biology, the idea is that, well, a lot of these things sort of interact with each other. There's going to be complex ways in which that interaction is going to produce novel phenotypes and different sort of circumstances. And so instead of just trying to understand things in isolation, you want to understand a system. And so how those variables interact with each other. And to me, that's just the simplest way to sort of think about systems biology, because we are emergent properties as human beings, right? And so how that comes about, I think, is a complex question that deserves more than just isolating those individual variables and understanding their contribution.
Right. Yeah, I definitely would agree with that. And I think especially when we're talking about very complex biological systems that are related to human health, that's really, you have to look at the interactions between all of the different cell types and tissue types, and in order to even start to model the overall system. And I mean, that's really what we're trying to do, right? Is develop the best models we can of functioning bodies and dysfunctioning bodies, and understand what differentiates them. And so, one of the approaches that is getting a lot of attention right now is this idea of digital twins. And I know you've done work with digital twins. So while we're in this sort of, defining terms phase, maybe you could talk about what does that mean? How does that work? How do you apply this concept of the digital twins in your own research?
Yeah, so the digital twins, the concept has been around for a while. And coincidentally, the concept early on was used by the aerospace industry. So like Boeing. And so the idea was they had jet engines, they had planes that would go out and they would have flight hours and do their thing, and they were scheduled for maintenance. And they would come in and they would notice that, okay, this plane is really close to having some type of mechanical failure and needs to be looked at. This other one with the same amount of flight hours is doing just fine. And so they realize that there's huge variability in the usage of the planes, and it's not just a single time, kind of measurement. Sure. It's more of sort of usage. And so because jet engines, they know everything about the physics of what's going on in there and the materials and stuff, having these additional measurements, they could essentially tell when an engine needed to be serviced based on those intermediate data points. And so the idea of a digital twin would be, okay, now we can simulate these, and we would be able to simulate, okay, when would these parts sort of break down relative to these others? And that was really sort of the idea behind a digital twin.
Got it. Yeah, and I mean, I think that makes perfect sense. And in some ways, that analogy of, it's not only about the time that a piece of equipment has been in use, but how it's been used, I think parallels very nicely with this concept of biological age versus chronological age.
Yes.
Of course, the difference is that we understand very well all of the components and the physics of airplanes or automobiles. We don't understand much of the biology of the human body, or really any animal.
Yeah.
And so how do you think about that in the context of trying to use this sort of digital twin approach in, biomedical research, healthcare? Kind of where are we now? And how do you deal with the large degree of uncertainty in these models?
Yeah, so that's a great question. So there's a book by David Epstein called Range, and in Range he has sort of a two class of problems. One is it's sort of a kind environment. So think about chess. All of the rules are explicit. There's only so many moves that you can do within chess, right? That's a kind environment, and that the defined rules are very clear. Then you have a wicked environment where not everything is known, and therefore, because you have, say, missing data, you have a missing understanding, and all of the different range of possibilities is vast and complex. And with that wicked environment, if you're trying to reverse engineer something, that's a really hard thing to do. So a big part of building digital twins, and a big part of what I work on, and along with my co-founders at Fulcrum, in particular Tom Patterson, who comes from the aerospace industry, is trying to figure out what we don't know with things. And so putting those puzzle pieces together, reconciling disparate data, is arguably the main task in building a digital twin.
So you mentioned Fulcrum. Can you tell us what Fulcrum is?
Yeah, so Fulcrum is a startup. I'm a co-founder of Fulcrum along with Tom Patterson and other colleagues of mine. And the idea of Fulcrum is to actually build a technology, a platform that models brain homeostasis, and with that model, be able to apply it in different ways, one of which is a digital twin, the main one being a digital twin. What you can do with a digital twin and what you can do with a platform also is almost like a Swiss Army knife of options of what you can do. So one of the things we've explored is how that could be a decision support tool for clinicians in understanding brain health specifically. Another endeavor that we're working on is also developing drug targets or interventions and therapies that would be able to address Alzheimer's and dementia.
And where do you feel like we are in terms of being able to have useful digital twin models of the human brain? Because it strikes me that the complexity is so enormous. And again, I'm not an expert in neurophysiology or neurobiology, but my perception is there's so much we don't understand. But that doesn't mean these tools can't be useful, right? And so that's I guess the question I'm asking is, are they useful now? Are they close to becoming useful?
Yeah.
What's your sort of take on that?
Yeah. So, don't let perfect be the enemy of good,
exactly.
Right?
Yeah.
And one of my favorite quotes too is from the statistician George Box. That is, all models are wrong, but some models are useful.
Yeah. I love that, and always try to emphasize to people that concept that everything we do in biology is simply trying to create a better model that more closely matches the reality of the situation. And models are useful, but they're also wrong, and understanding where they're wrong makes them more useful.
Yeah, exactly. And so we like to extend George Box's quote and say the models that are most useful are the ones that can reconcile the most data. And so that gives you the more most complete picture possible with what you have in terms of the model. And to sort of answer your question, we have built a model of brain homeostasis. The other concept that I think is a really important one with digital twins is that model becomes sort of like institutional knowledge, right? Where it is a repository of understanding, and it's not like a one-off thing and then you just build something completely different. No, you learn something from that model, and you have parts of it that, it's a very large, complex model, just like human physiology and is very complex. And so you can update that model as you learn new things. You don't start from zero. If anything, the challenge is going to be part of reconciliation of, okay, these parts work really well. You have sort of proxy measurements to say, oh, yes, we've got this aspect of the biology right. We can look at these things in the blood. These other things maybe we're not so clear on. So then we get new pieces of information, new pieces of data. Okay, now how do we make those work with the existing ones? And that is a very difficult process in building a digital twin. But that is also, that rigor is really important, because that then goes to the whole idea of reproducibility, right? Yeah. And so the job of reconciliation is arguably a main focus of what you're trying to do when you're building a digital twin.
Right. We should have probably done this early on, but do you have a concise definition of digital twin that a non-scientist would get? Or are they metaphors that you can use to?
Yeah, we can try. I mean, another challenge similar to the concept of systems biology is the way people use digital twins can vary dramatically. So one of the more common uses that is out there now is using a digital twin in, say, like a clinical trial. And the idea is if you can estimate sort of like effect size and the variability you're going to see within a population, then you can arguably downsize or reduce your sample size, particularly for the controls, which would save you money. Sure. And so a lot of people are sort of using digital twins in that sense, or they're calling them digital twins to basically just inform trial design. But a definition of a digital twin, for me, it goes back to the aerospace industry. What is your goal with that? And for us, it's trying to sort of emulate human physiology, brain homeostasis, in a way that is going to be informative. Another thing too, going back to the aerospace industry, is they can measure just about anything in a jet engine. We can't measure everything, but one of the concepts that is important for a digital twin is what you can do is you can come up with synthetic variables of human biology that say, I can't measure this aspect of human biology directly, but I can come up with a proxy measurement or a synthetic variable that does measure that. So another goal of sort of digital twins is to have a representation of something that you can then model and have an understanding of how say permutations reverberate throughout the system.
Mm-hmm. And when you say you are trying to develop, or you have developed a digital twin or a model for brain homeostasis, what does that actually mean? Like what What goes into that?
Yeah, what goes into it? So this is really the work of my colleague Tom Patterson, who comes from the aerospace industry. He was also, there's a discipline called quantitative systems pharmacology that is now in almost every big pharma, they have sort of a division that does QSP, and they are the modelers. And so Tom is actually a pioneer of part of QSP. And so he's again bringing this sort of discipline, which I think it's fair to call it a discipline, even though it doesn't really exist as a discipline writ large, but it definitely does these principles. It is a very principle-oriented approach. And so, again, going to a digital twin, what I would define as a digital twin, one way to sort of think about it is that if you want to model interventions on an individual, you can create a digital twin from their data to say, okay, this is the best representation. This matches what your data looks like.
Mm-hmm.
And then
and their data could include blood work, genetics, multiomics,
yes.
if you have it, basically whatever you can measure
yes.
would inform the model for
right.
their digital twin.
Right. And there's a few ways to sort of think about this, but I think you're hitting on something that's really important in that you can collect data on somebody. What I like to think of as a digital twin is not just a statistical association or understanding of what that data represents, but how do you tie that into the biology?
Mm-hmm. Right. Absolutely. Yeah.
Yeah. So if you're modeling, say, cerebral perfusion, you want to be able to, and the things that impact cerebral perfusion, then you want to have within your model ways to tie that in, anchors to tie that in. And to me, that I think is, if we're going to get into sort of a pure definition of a digital twin, I would suggest that that's going to be one of the more useful ones, because that is closer to, in terms of abstraction, closer to what you're trying to do in trying to understand a human physiology.
Well, that's really helpful, and I think one of the questions that I wonder about in this space, again, this is getting to where kind of where are we at now, and where might this get to, is when you talk about brain homeostasis, like that's a big thing, right?
Yeah, it is.
But I'm still sort of like, what is that? I mean, I get what it
Yeah.
means in the sense of like your brain is able to maintain function within some boundaries of good function.
Right.
That's homeostasis, right? Ability to restore
Right.
function when perturbed. But but that's made up of a bunch of smaller things, like even cerebral perfusion.
Right.
And, there's all sorts of specific factors from the subcellular to the cellular to the multicellular tissue level.
It is a multi-scale problem.
Yeah.
For sure.
And I guess what I'm wondering is, when you design these digital twin models now, are they very focused on specific cellular processes, or like how comprehensive can they be at this point?
Yeah, so the platform that we have and the model that we have does go multi-scale, in that there are specific things in there that represent, down to the cellular level, up to the physiological level. And again, those spanning those scales is a big part of that. And one of the things, I guess, to help sort of ground an understanding of what we're doing with the modeling. So ultimately, it's all math. It's all ordinary differential equations in there. And but what it does on top of that is that you want to be able to relate, say, that cerebral perfusion to what is happening over decades, right? And so how do you sort of reconcile those things and represent them within the biology? And so you can do that in a number of different ways. For us, one of the things that, when we talk about homeostasis, what we think, what you decide to model also sort of is an important decision, right? In terms of the pathology, we model some things. You can model just about anything you want, but is it going to be relevant for human disease and neurodegeneration? So one of the things that we've chosen to model is cholesterol homeostasis,
right?
Because that is, I think, we again, through this process of building these digital twins, building these models, what it forces you to do is you have to take a lot of assumptions and make them sort of explicit in the model, right? So there's some guiding principles that we have used in building this model of brain homeostasis. One of which is feedback loops, and this is, again, so going back to systems biology. And my background was really looking at transcriptional regulation. Well, a lot of the tools in models that I was building for transcriptional regulation were essentially acyclic directed graphs, meaning that they are sort of like dominoes. And you have a starting point and sort of, cause and effect, ending kind of a thing. And I was very unsatisfied because if you think of a transcription factor that, say, regulates the expression of another transcription factor that's then regulating other genes that then are going back to regulating that transcription factor, you've got these loops, right? And the tools for those feedback loops are really not very good within systems biology, arguably. And so I was sort of frustrated with that. And one of the things that was really attractive to me about this idea of modeling with digital twins is the emphasis on getting the architecture right of the feedback loops. And so, so much is again, that abstraction. What do you care about? What does the brain care about with respect to cholesterol homeostasis? It has a lot of different feedback loops around cholesterol specifically because it's so important to maintain proper cholesterol levels, at the cellular level.
Right. Yeah, that makes sense. And, I think one of the other questions I think about a lot with these digital twins is how do you know how well the model is working, right? I mean, obviously you can ask, how well does it match up with measured data, right? But sort of beyond that, like what are the tools you can use to understand, where it's working well, where it's not working well, how well the model is performing, and particularly in something like, when you're looking at brain homeostasis, which, as you've alluded to in humans is a multi-decade sort of process of breakdown. Like how do you test the model, right? Because fundamentally that's what we try to do in science, right? Is break our models and make them better.
Yeah. So that is still an ongoing endeavor of how do we test this model, how we demonstrate that this model is correctly representing human physiology, and I'd say we're still working on that. But you mentioned reproducing data. So for example, there's multiple levels of data that you can try to reproduce. So maybe on the highest level, as ApoE is the gene that is most highly associated. E4, right. ApoE4 is most commonly associated with Alzheimer's disease. You have E4, E3, and E2, and you can look in the literature, and there are Kaplan-Meier curves of Alzheimer's incidence for each of these different alleles, right? And so one of the things that we do in order to sort of test our model at a very high level is can we reproduce those Kaplan-Meier curves with those individuals? So like, okay, we're going to create a digital twin. We're going to expand that. We have a digital twin. We're going to create a digital population of a million individuals. And when we sort of let those simulations go, do they break? And we assign them to different ApoE groups. Right. Do they reproduce the types of biomarkers that are found to be associated with ApoE3, ApoE4, and so on? Right. And so that's interesting too, because at this point, your simulation, again, it's very rooted in the biology, but you're then trying to reproduce population statistics around, say, the distribution of amyloid beta or the distribution of phospho-tau and so on. And so being able to get all of those things right can be a really tricky thing,
yeah,
right?
Especially when there's in the real world, all this additional environmental complexity. Yes. Yeah. I mean, it seems like a very daunting challenge, but those are the best kind to go after sometimes.
They are. And again, one of the principles too that I like to sort of articulate with this is that a lot of times in science we get very focused on precision. Yeah. Can we get this down to the 10th decimal place, right? With a digital twin, particularly when you're working with a vast array of data, you have a different mentality or approach, and it's more akin to the way like a GPS works with satellites and triangulation. Right? What you want to do is you want to get orthogonal measurements from different locations and use that to triangulate your understanding of what the location is for something. It's the same concept within the digital twin. When you've got so many different data types, you're less concerned about the absolute precision of getting this one thing particularly right. You want to understand how it relates to the other things to produce something that you can measure that is the combination of all of it.
I like that analogy. That's really helpful to sort of put it into perspective, a different frame for how you think about the problem. So I want to get into Alzheimer's disease here in a minute and go a little deeper on that. But when you're talking about your model for brain homeostasis right now, is it primarily focused on Alzheimer's disease as the readout, or can it map all sorts of, because Alzheimer's disease is obviously only one specific type of brain dysfunction that happens in people with age. There are a variety of other ways that the brain undergoes different types of pathology, manifest as different diseases, or in some people just manifest as age-related cognitive decline. So does the model try to pick up on all of that, or is it really focused on Alzheimer's at this point?
Right now we are focusing on Alzheimer's, but it is a brain homeostatic model, right? And so for that reason, the aspects of the model that we build out are ones that are currently focused on Alzheimer's, but we absolutely would not take much to turn our attention towards, say, multiple sclerosis or Parkinson's disease or other neurodegenerative disorders or Lewy body dementia, because a lot of the same biology is occurring in those neurodegenerative disorders as well. So,
yeah. Okay. So let's dive into Alzheimer's then. So sort of just kind of big picture, like I think everybody realizes this is a huge societal problem, right? And has immense economic burden, social cost for patients and caregivers and family members. So from your perspective, I mean, I think a lot of people are frustrated. Like, why hasn't science figured this out yet, right?
Yeah.
Wow. So why hasn't science figured it out yet? What are the barriers that have really, I think, stymied progress, at least in developing effective therapies for the disease?
Yeah. So I mean, I think there are a lot of ways to answer that, and I don't know that that's a systems problem right there, systems question right there.
Right.
They can go all the way to funding agencies and so on.
Yeah. Let's leave aside like the policy fund. I mean, we all agree more funding for science would be helpful in a lot of ways, a lot of areas. What makes it a really challenging scientific problem to solve?
Yeah, so I think scientifically, it's a disease, neurodegeneration. So a couple principles. Things start to sort of go off the rails, decades before any symptoms manifest, right? But the other thing to keep in mind is the brain is really resilient. It has a lot of adaptability, and because we are so good, and arguably evolution has selected for this, we need our brains to function. Right. And so when things are sort of slightly going wrong, we're really good at compensating, and we're really good at hiding some of those deficits that and even if we're not working if the brain's not working optimally, it's still working pretty well. Right. And so because that has been that is masked for many years, it's really hard or it has been hard, I think, to sort of understand the early events that happen that sort of contribute to that and identify them, across a population, particularly because they're also so diverse, right? So, as a complex disease by definition, it's not just a single gene. There are, at this point, there's, I think, over 100 different GWAS loci, genes that are potentially implicated in the disease. And then you throw on all the environmental factors that are also contributing to things. And you've got quite a diversity of etiology across a population. And so Alzheimer's disease, it was defined by Alois Alzheimer in like 1909, I think it was. And he actually saw it in a woman who I think was in her late 50s, which would now qualify as sort of early onset Alzheimer's disease, right? But he saw the pathology, he saw the plaques, he saw the tangles, he even saw the lipid droplets in there. And so one of the things that I think has also been a challenge within Alzheimer's disease is defining what is the disease, And so if you can't agree on that, it's really difficult to pinpoint a specific etiology, especially when that etiology is going to be different across the population.
Yeah. And I mean, related to that, do you think that the way that Alzheimer's disease was initially and maybe still clinically defined played a role in the false starts, maybe is one way to say it, in terms of what people focused on?
For sure. So I think that there's a governing body, and I don't remember the name of the governing body, but you would not actually be able to diagnose somebody with Alzheimer's for many years until it was postmortem, and you could actually go in and see
you had
pathology.
to actually look in the brain. And what were the clinically defining features?
Yeah, these were again the plaques, the tangles, and the lipid droplets.
Right.
And then there's been a long history of how do we define Alzheimer's. There's the ATN sort of criteria, which include also cognitive testing, as well as the biomarkers and stuff that can be associated. And so really you have somebody that might go to 3 different clinicians the definitions of, or where they are within the disease can differ substantially. Or there's some variability there. So that's again been a challenge if you can't even agree upon what is what is this patient.
And we may get to this, but I think there's even a question of, is it a single disease, right?
Yeah.
So it's
for sure.
I think there's still a lot of lack of clarity around what Alzheimer's disease is at the molecular level, for sure. And even, as you said, there can be differences in terms of what the clinical presentation looks like, right, and how it's graded. So given that, getting back to this question of why has it been so hard, I heard a statistic like there's been 400 failed clinical trials. So it's not as if there haven't been a lot of shots on goal. Why haven't we gotten better Alzheimer's drugs, in your opinion? Yeah,
so this is, again, I think it's a complex question, and I can identify what I think are some of the contributions, but it's difficult to say. I mean, one of the things too is the focus on amyloid, right, for decades, I think has been a challenge, and I think it's interesting too because now we actually have the monoclonal antibodies are really good at removing amyloid.
Right, you're talking about the newly approved class of drugs
yes.
that are targeting amyloid directly, and can reduce the plaque burden in patients.
Yes.
Right.
Yeah. In fact, on some of the latest studies, they work so well that the individuals are no longer qualify as being amyloid positive halfway through
so
the
trials. So they don't have Alzheimer's disease anymore, based on at least that definition.
According to that definition, yeah. And so I was at the AAIC, which is sort of the annual Alzheimer's meeting in Toronto this year, and there was people who were presenting, so for the first time, individuals who have been on these drugs, and they do the pathology afterwards. And yes, their plaques are reduced, but they're not gone. And so I think this, again, the field is still learning on this because what you're measuring again from the blood or from the CSF, you might see, it might be below a certain threshold, but are you completely getting rid of the plaques? No, you're not, according to the pathology. Yeah. But I still don't think that means that we just need to be much better at getting rid of the plaques.
Well, that's what I was going to ask. Yeah. And what is the current sort of consensus on how effective those approaches are in terms of functional improvements in
so
patients?
They've done the clinical trials and they do see an improvement, and I think that depending on how you do the math, the monoclonal antibodies currently can give you the equivalent of about 6 months of dementia-free life, or extend it an additional 6 months.
And does it so again, for people who are already, presumably most of the people who've gotten these drugs have some significant cognitive deficit before they go on the drugs, right?
Yes. Well, this is,
I guess the question is it just giving you, is it just slowing? I mean, I understand there's a little bit of improvement.
Yeah.
That's my understanding. Maybe it's more than a little bit. That's kind of why I'm asking.
Yeah.
Or is it really just kind of stretching out that last phase? Right?
Yeah. So if you look at sort of the trajectories of individuals, there's a slight improvement, particularly early on. And again, the way they design these trials are typically trying to get people that are in MCI or early stages. And so you see an improvement, and then the slope is pretty close to the same one of those who don't have the drugs.
Wow. Okay.
And so this is as a researcher, one of my interpretations, and again going back to this modeling effort that we're doing with digital twins, is that what that says to us is that you're not really doing anything to address the underlying cause of the disease. So I talked about the guiding principles of digital twins, and there's the feedback loops, but another one that we do is mass balance. And so one of the things that we have noticed in particular with these monoclonal antibody therapies is what they have seen is that the brain continues to shrink in size. So you continue to seem to lose neurons even as you're reducing the amyloid. Right. And so this is, again, we think is sort of evidence that you're not treating the root cause. You're still getting neurodegeneration. You're just getting rid of some of the amyloid, which is probably beneficial. And in fact, going back to how might that be beneficial, one of the things that we think is going on when you remove those plaques, that's actually increasing cerebral perfusion. Because a lot of the plaques also contribute to CAA, which is going to reduce cerebral perfusion. And
maybe just define CAA.
Yeah, it's I can never remember what it stands for, but it's amyloid angiopathy, and I can't remember the cerebral amyloid angiopathy.
Okay, you want to just say it? We'll cut all that. Like I don't get.
Yeah, cerebral amyloid.
I'll just say that again. Maybe just define CAA.
CAA is cerebral amyloid angiopathy, and so it is a known thing that impacts your cerebral perfusion.
Got it.
Yeah.
Yeah, and I mean, I think so. Most people are probably by now familiar with the idea that the amyloid hypothesis is incomplete, right? But maybe it's worth just sort of very high level. You tell me if I've got this right. Like for many years, decades, the concept was that the formation of these amyloid plaques, the tangles, were causing neuron death and Alzheimer's disease, right?
Yeah.
That's what you were saying.
Mm-hmm.
I think we've now learned if they are a causal factor, they're not the only causal factor and potentially not the primary causal factor, because if they were, then getting rid of them should at least stop the neuronal loss. And you're saying that's not what is seen. And I know your several models have started to be proposed for other, causes of the neurodegeneration. I know your group has developed one related to cholesterol metabolism. So I want to dive into that and maybe start by telling us, how did you get on that path, and then take us through the model that you guys have come up with.
So again, going back to mass balance, right? So as I was saying that, if the brain continues to sort of shrink, well, how does the brain get rid of that debris? And, cells, particularly the brain, has, high amount of cholesterol. I think 20% of the cholesterol in your body is in your brain. And cholesterol is really important. Your brain is also running really hot. It's only 2% of your body mass, but it uses 20% of your energy. And so it's running hot. And cholesterol, as I mentioned, has a lot of feedback loops. It's really important. And one of the things that cholesterol really impacts is cell signaling. And this is because it impacts, membrane fluidity. And so the extent to which receptors are able to function on cells is going to be a function of that membrane fluidity. And cholesterol is a key component that contributes to that fluidity. So if you have too much cholesterol or too little cholesterol, you're not operating in the space that evolution has sort of designed most of these receptors to sort of function. And so for that reason, you're going to screw up a lot of the cell signaling pathways that are going on. So in that sense, that's one of the reasons why we're thinking of cholesterol. But again, going back to, the brain's it's in a neurodegenerative disorder, you're losing cells. You've got to be able to process those. And another key thing with that we know about Alzheimer's is when we look at the genetics outside of ApoE, which is, by the way, a lipid transporter.
Right. So
again,
associated.
very much on target,
right.
in terms of trying to understand why is cholesterol so important.
Yeah.
The other genetics within Alzheimer's, a lot of them point to microglia. Right. And microglia are a cell type in the brain that are responsible for clearing the debris. That's one of their jobs. They have many jobs, but that is one of their jobs. And so when with aging, you have neurons will die, and just part of aging is sort of a slow baseline loss of neurons. But the extent to which your microglia can handle that debris, they can stay up on it. They're essentially the garbage trucks.
Right. I mean, microglia are like the component of the immune system in the brain that's responsible for clearing out garbage.
Yeah. Clearing out the debris. Yeah. Another one that they're responsible for is myelin turnover, right? So there's going to be a lot of turnover, and with aging too, the amount of turnover that happens increases with age, right? And then in MS, when you have sort of episodes, you get a lot of demyelination. So again, neurodegeneration, very common.
Yeah, and I mean, most of the time, I'm sure people have heard of neuronal inflammation or brain inflammation, and generally that's referring to activation of microglia, right?
Yeah. So they absolutely are the sensors for inflammation, right? And one of the things too that amyloid will also contribute to is that inflammation. Yeah. So there definitely is a relationship there. So the microglia, what they do with that debris is they phagocytize the debris, they take it in, and then they've got to process that debris. And the question is, okay, where does it go? And so this is where ApoE comes in, because when microglia ingest the debris, then there's several different microglial subtypes, but there's one that it produces ApoE. And then that ApoE is used to package ultimately that cholesterol debris in lipoprotein particles. And those lipoprotein particles are then, when you sleep, they are excreted out of the CNS.
Right, which is, again, this idea that sleep is important for clearing out the brain.
Clearing out the debris, exactly. Yeah.
And so is it the case that the E4 allele, the allele that is associated with Alzheimer's disease, is less effective at clearing out that debris?
It's less effective so there's a few different qualities of it that sort of make it less effective, one of which is its half-life. So it has a much shorter half-life. And so we think about this in terms of its carrying capacity. The amount of debris that it ultimately can sort of help take out of the brain is less than what an ApoE3 allele can do. And that's partly for the half-life, but also some of the binding dynamics of it as well in terms of the size of those lipoprotein particles. There tend to be fewer of them, but they're larger.
Okay.
Yeah.
So let's come back to the model that you guys proposed. Maybe can you just give us kind of a high-level overview of what you think is going on here?
Sure. Yeah. So we talk about this in terms of a unified hypothesis. But again, going back to Alzheimer's disease is not a monolithic thing. But in terms of the underlying biology, what we hypothesize is that it really is a challenge in debris management. And so when you have those baseline, the debris that sort of accumulates with age, there's a rate at which they can be cleared by the microglia. When that rate is exceeded by the microglia, then the microglia then signal to astrocytes that say, hey, we need some help. And at that point, those astrocytes become phagocytic as well. So then they are recruiting more to clear out this excess debris. We can come back to it, but you can think of, say, all of the risk factors that are known to be associated with Alzheimer's that might increase the amount of debris. Something like a traumatic brain injury, or if you're exercising and you have better cerebral perfusion, that might be less, or things even like COVID or other sort of things that might sort of increase the rate of debris generated. So going back, so once the microglia, their capacity has been exceeded, they recruit the astrocytes. Astrocytes, one of their main jobs is to actually support metabolically the neurons. And so there is a relationship between the astrocytes and neurons. So astrocytes, it's called the astrocyte neuron lactate shuttle, ANLS. And neurons, similar to the way, one of the reasons we have batteries is neurons outsource their energy to astrocytes. This is what helps make certain neurons be able to do whatever they want to do on demand, right? And so glycogen in the brain is stored within astrocytes. And so when neurons need ATP, what they do is they signal to the astrocytes, say we need more lactate. That lactate is then shuttled over to the neurons, and that is the basis then that goes into the Krebs cycle, and that produces ATP. And that's a very quick way for neurons to produce ATP for long-term potentiation and sort of do their thing, right? So now let's go back to when those astrocytes become phagocytic. What they're now doing is they're taking in cholesterol debris. And part of our hypothesis is that when that debris is coming into those astrocytes, one of the things that it does is it raises the overall amount of cholesterol in the cell membrane of those astrocytes in a way that is then disrupting the signaling that is going on between the neurons and the astrocytes. So when the astrocytes receive that signal from the neurons and say, hey, I need more energy, the astrocytes can't hear it because you're no longer in the sort of optimal space for that signaling pathway to function, the GPCR.
So did I understand correctly, in this case, cholesterol dysregulation is affecting both the microglia and then the microglia change the function of the astrocytes.
Yes,
to
they
become
recruit the
phagocytic.
astrocytes. Yep.
Cholesterol also then affects that function, so then the astrocytes can't support the metabolic health of the neurons.
Yes.
And then that presumably
creates
is going to
more
lead
debris,
to,
right?
It's a vicious cycle.
Exactly.
Driving neurodegeneration.
That's right.
Okay.
Yes.
That's very cool. How does that fit with some of the other models that are out there? One that we hear about a lot is the sort of pathogen-induced Alzheimer's disease, whether that's viral or bacterial antigens in the brain, or maybe even bacteria in the brain. Does that fit with this model?
Yeah.
Is it inconsistent?
I think it does. So that's one area. So I actually do some research on the role of herpes viruses in Alzheimer's.
Right.
Yeah. And so that is something we have not yet built that into the model, but the place in which it ties in biologically, I think, is we have a good idea of where that is. And that's similar to the inflammation side. So one of the things when when microglia encounter pathogens, what they do, you're sort of activating, PI3 kinase and some of these other pathways of inflammation. What that actually does too is changes where you're stashing away your lipids. And I think part of this is because viruses in particular, they need cholesterol to help make their the envelope of the virions and stuff. And so they want to sort of take the resources away. And I think one of the responses or ways to sort of prevent that is to sort of stash away the cholesterol. Where do you stash it? You stash it in the lipid droplets, right? And so when you're activating TLR3 pathways and so on to do this, then you're getting a change in the distribution of that cholesterol. And so,
I see.
Yeah, so that's one way in which we think is sort of happening.
And so presumably anything that drives that type of inflammation could feed in at that point, right? Leakage of the blood-brain barrier, or really even mitochondrial dysfunction that drives inflammation.
Yeah.
And then what about this idea, you hear people talk about Alzheimer's as type 3 diabetes. How does that fit into this model?
Yeah, so I think that goes back again to the sort of metabolic support for the neurons. So a couple ways to sort of think about this. With the ability for the astrocytes to metabolically support the neurons, anything that is going to sort of reduce the amount of available energy for those neurons, I think is going to make them at a population level sort of more susceptible. So particularly again with sort of aging. So. And this is actually what is known about tau phosphorylation, is that it's directly related to ATP deficits. So this is a complex problem. Like you can think about okay, you're trying to do maybe a hard math problem. It's making your brain hurt. Well, what you're actually doing is you're increasing cognitive burden on your brain, and you're saying, hey, I need more assistance in doing this. And so that is upping the amount of ATP that you need to essentially solve the problem. And so when you are doing that and you aren't able to metabolically support those neurons, then you're going to run an ATP deficit. When you have an ATP deficit, then that is going to contribute to that triggers sort of that's calcium dyshomeostasis kind of a problem that then causes phosphorylation of tau. Right. And so this is how those things we think are related.
And I think this is really nice because now you can start to think about multiple points in this cascade where you could potentially intervene. Right. And I want to come back and talk about interventions in a bit, but it also starts to explain why, if this is all correct, if you're intervening, down towards the end of this process, even though it's a network, I'm just going to think of it like a linear process because that's the way my brain works, at the plaques and tangles, why you would get sort of more modest outcomes than if you could do something that's more fundamentally upstream or earlier in the cascade.
Yeah, exactly. So things that you can, this is again where some of the risk factors we know about, like exercise. What are you doing? You're increasing your cerebral perfusion. You're increasing the amount of nutrients. You're metabolically supporting
probably energetic efficiency as well. Right?
Absolutely.
Yeah.
Absolutely. Yeah, for sure. It's like any of those things, or and diet too. Like, what is your energy availability going to be with and again, this is a personalized thing, right? Because some people can do better, more so on carbs versus fats or so on, kind of a thing. All of that sort of can factor into how well are you able to metabolically support your own neurons.
So one of the thoughts I have is, this is a very appealing sort of, as you've described it, unifying model of Alzheimer's disease. But, presumably we know that many of these processes are happening during normative aging, right? So do you think of this really as an Alzheimer's disease mechanism or a brain aging mechanism that in some people, for a variety of reasons, leads to Alzheimer's disease, but in other people leads to other brain changes? Is that
absolutely
correct?
I think that's a very good framing. Yeah, and I think how to distinguish normal aging from pathological aging towards neurodegeneration can be kind of a slippery slope.
Yeah, and I would even say, I think it's sort of arbitrary in some ways to even try to frame it that way.
Sure, yeah.
Because certainly there are aspects of aging that are not pathological, but when we talk about functional declines, sort of by definition, if it's a functional decline that impairs homeostasis, that's pathological, right?
Agreed.
But it can manifest itself in a variety of different ways in different individuals. Right. And this is another thing that I think about because, my background, I've worked in, dogs with the Dog Aging Project. Dogs get dementia. They don't get exactly Alzheimer's disease, but they get dementia, and lots of studies in mice and even simpler animals. And I think the way people talk about it is as if Alzheimer's disease is a human-specific disease. Maybe there are some non-human primates that sort of get Alzheimer's, but that's more based, I think, on the plaques and tangles and some of the specific neuropathological hallmarks. But is it your belief, and maybe you've got evidence that this sort of more fundamental underlying mechanism is in fact going to be more broadly shared across mammals at least, and maybe that can account for dementia-like conditions in other animals that aren't Alzheimer's disease?
Yeah, so this is actually one of the reasons why we think the mouse models are not very good.
Oh, interesting.
Tools.
You're talking about the models where people try to engineer Alzheimer's disease.
That's right.
In mice.
Yeah.
So tell me about this.
Yeah. So we sort of, mice, most of the Alzheimer's models, and I don't even want to call them those because it's more of some of the Alzheimer's pathology associated with.
And maybe it's worth just stating explicitly what is typically done in these mice.
So these are all transgenic mice that typically have 2 or 3 familial mutations, meaning that these are rare mutations that have been discovered in humans to be associated with the disease. So typically, amyloid precursor protein, which is ultimately making the amyloid.
And these are typically early onset.
These are early onset, and I think that
people.
Absolutely, and I think it's again a very important point that these are early onset. So they'll do those. They'll humanize the tau. They'll put in human ApoE4 in there as well. So they're really sort of stacking the deck towards this Alzheimer's pathology.
Right.
But one of the things that we think is really missing in these mouse models is, again, that sort of ramp up of debris that you get over aging, because mice, they only live for a few years. And I think there are ways to potentially create better mouse models in which you are recapitulating some of these same sort of aging and accumulation of debris.
Yeah, they're called aged mice.
Aged mice. Yeah.
I mean, so I love it because I've been pounding the table about this for, it feels like decades. It's probably been at least a decade that I think one of the reasons why most of the Alzheimer's models in mice fail to recapitulate therapies in humans is because they're young mice engineered to develop some of these Alzheimer's pathologies. If you did that in aged mice, I think you'd have a better chance. And this is a good sort of biological explanation for why that might be the case.
Exactly. Yeah. That's exactly what we think.
Yeah. That's super interesting. And again, I'm fascinated by the dog situation because dogs do develop this dementia syndrome, canine cognitive dysfunction, which, I think the statistics are maybe 15% of dogs will get this during normal aging. So it's a, fairly significant fraction of the population. And might this sort of a mechanistic explanation be at play? It'd be fascinating to study.
Agreed.
And sort of related to that, I always wonder, and I'm sure you've thought about this, what other human brain diseases might this mechanism play a causal role in? And related to that, why then do you get the different phenotypic or clinical presentation in different people?
Yeah, so it's a good question, and it's a little outside of my area of expertise, but I think some sort of high-level answers to that, again, going back to multiple sclerosis, right, which is where you're going to have events where you have severe demyelination in a specific area. And then those microglia have to do the same processing for all of that debris. So that's going to be sort of an acute focal, and that's where that happens in the brain is also going to sort of matter in terms of the pathology. Another one, Parkinson's, as we discussed before, that's also in the substantia nigra. You're essentially getting demyelination of some of those same neurons, which, by the way, have very thin amounts of myelin or very low amounts of myelin to begin with, so that they're already sort of more susceptible. So it essentially could be some of the same pathology, but where it's happening in the brain is what determines what the presentation is for the disease. Right.
Yeah. And do you know, or is there much known about how this sort of cholesterol metabolism differs in different regions of the brain that could make certain regions more or less susceptible? Yeah.
So I don't know. I don't think we have a good understanding of that now. I think some of the things that we think about as might be more about the energy demands of the cells. Sure. Right.
Yeah, that makes sense.
And so some neurons, and this has been shown, some neurons are going to be more susceptible because they run hotter. They're firing more often, right? And this also, going back to risk factors, we think about anxiety as associated with Alzheimer's, right? If you have more synaptic firing, then you're sort of using up more energy. You're going to be more susceptible to energy deficits. And that is, again, going to be a problem in terms of creating that neuronal debris, dying neurons. You're going to be skewed a little further on. And that's true about, again, for almost any risk factor.
Yeah, that makes sense. Yeah. Okay, so let's talk about interventions. And we talked about kind of what the status is of the current FDA-approved drugs, which, it's good that we have them, I guess, but they aren't game changers. I think most people would agree with that. So how does this model, I guess we'll talk about specific interventions, but in general, like, how has this development of this model changed your thinking around what's likely to work, what's not likely to work? What are some strategies that could be used to find new ways to target Alzheimer's disease?
Yeah, so again, going back to the feedback loops and, again, the cholesterol homeostasis, if you're going to have a therapy that is only going to, say, move the cholesterol from one area to another and ultimately not help that cholesterol get out of the brain, then that arguably is not going, it might buy you some time. It might be effective, but it's going to have a ceiling or a limitation in how well it can do that. Also, those things that sort of contribute to that neuronal death too, if you're not addressing some of those specifically, then sure, you can improve your ability to handle the debris and get it out, but you're still losing neurons, right? So these are some of the sort of principles in terms of devising a therapeutic strategy that we're thinking about. Again, with the whole idea of how do you maintain homeostasis? How do you maintain, energy to sustain those neurons, to keep them alive, so that you don't sort of lose them at a rate faster than you want to?
Yeah, and I mean, that makes perfect sense. I think maybe the way I initially think about this is, right, there are some obvious places in this cascade that you could probably target that, like you said, wouldn't fix the root problem.
Yeah.
But would greatly delay or slow the decline. Yeah. And again, maybe to the point where it becomes, sort of like prostate hyperplasia in men, where you slow it enough that it's not going to be a quality of life limiting factor,
right?
At least right now. Those might be easier places to target.
Agreed.
But I agree with you that the closer you can get to the root cause, the more effective the therapy is going to be. And we know lifestyle can have a huge impact, right? In progression towards Alzheimer's disease and brain aging in general. Maybe it's worth just touching on, how do you think about diet, exercise? You talked about sleep, but maybe just a little bit more explicitly sleep. And you also talked about stress. I think that's really interesting. And so maybe we can touch on each four of those and how they fit into this model. So let's start with diet. And I mean, to the extent you can, I get there's a lot of speculation here, but to the extent you can, how do different dietary strategies, approaches, or even just at the level of quality of diet kind of impact the way you envision Alzheimer's disease risk through the lens of this model?
Yeah. So again, I think anything that goes back to, are you metabolically supporting yourself sufficiently? And with diet, as we know, with processed foods and sugar, what you're going to do is you're going to have big spikes, in your blood sugar. Yeah. And in terms of sort of maintaining homeostasis and expectations of having that glycogen in your astrocytes to support your neurons, I think we know that that's not going to be beneficial, right? So, or to the extent that you can have a healthy diet, and again, I think that there is personalization. I think really matters in this case, right? Because some people are better at processing carbs than fats, right? Sure. And I'm actually one of those people. I'm a fast twitch person, so I'm homozygous for ACTN3. And I actually notice this. So I'm more susceptible when I eat higher fat foods. It's harder for my body to actually get rid of those fats because I have a lower mitochondrial density.
And you notice that just in the way you feel, or you've actually looked at biomarkers? And
well, I know this from 23andMe why I have that. Yeah. This is sort of my best explanation for what to have my own sort of brain fog.
Right. Have you ever done anything like continuous glucose monitoring or something like that?
I've not. I really would like to do.
All right,
I should do that.
We can help you with that.
Yeah, that'd be great. Yeah.
Yeah. No, that's a super, super informative tool for those sorts of personalized responses to different types of food. But I mean, it sounds like, one level is just if you can maintain metabolic health of the entire body, that's going to maintain metabolic health of the brain.
For sure.
And we know, in general, strategies that can do that.
Yes.
Broadly speaking, across the population, right? Avoid highly processed foods, and in general, avoid added sugars. And then it gets to the point of individually trying to dial in for you what's gonna be optimal.
That's right. Yep.
Are there any strategies that you feel confident or comfortable speaking about that might be particularly beneficial or detrimental to brain metabolism, or is it really more at the level of whole body at this point?
I mean, I think they all contribute. I mean,
I'm thinking like, people talk about ketogenic diet or ketone esters in particular, and ketone bodies being a fuel the brain can preferentially utilize. I think the data there is intriguing. I'm just wondering if you've seen anything that makes you bullish or not so bullish about that as a therapeutic strategy, for example.
Yeah, I think there is good research. The Mediterranean diet for long-term for longevity is good, right? And I think going into a ketone diet, I think it's interesting too with that. So in terms of, again, cholesterol sort of metabolism, what's going on in your periphery is not necessarily what's going on in your brain, right? And there's you have the blood-brain barrier. But even I think this gets into sort of statins as well.
Yeah, I was going to ask about lipid-lowering drugs, and so we can talk about that now. Because I mean, one simplistic idea would be, well, if cholesterol is the problem, let's just knock cholesterol down to the floor. Right?
Yeah. And in doing that in periphery, so again, going back to feedback loops and mass balance, well, where are you putting that cholesterol? What is it doing? What form is it in with those statins? And I think that also sort of depends on which type of statin. There's, the lipophilic and the hydrophilic ones. There's two different classes of statins. And then I think that also depends on a person's genetics, because if we're thinking about feedback loops, where are you creating a bottleneck? Are you shifting some of that cholesterol from one area to another? I think that really sort of matters. And so I think there's mixed data out there, particularly on statin use for Alzheimer's. And I think some sort of suggest that it improves. I think that in some cases I've heard where it might actually make things worse. And so I think it's difficult at this point to sort of say. And what I would suggest is that a challenge with that is like, okay, you have to think about this in terms of homeostasis and feedback loops and mass balance. And I think a lot of the sort of models that people have out there are not necessarily rooted in those things.
Yeah. So let me ask you from a pragmatic perspective, right? So I think what you said, and correct me if I'm wrong, but I'm pretty sure what you said was that what you measure when you measure cholesterol and HDL and LDL in your blood isn't necessarily reflecting what's happening in the brain. And the question then is, are there good biomarkers right now to understand what is happening in the brain? Because we always measure it from blood, right?
Yeah, right. Yeah. So this gets into some of the research that we've done, where we have. So there's a large longitudinal data set called the ADNI data set, and on that data set they have multi-omics. So they had CSF proteomics, and they also had plasma lipidomics. And so one of the analyses that we did was we looked at the correlation structure of the relationship between those proteomics and those lipidomics. On top of that, there's also in ADNI, they do an assay called the Nightingale assay, which is basically doing a particle size of all of your different lipoproteins. Right. Yeah. And so there's certain lipoprotein particles, the XL HDL ones, that almost exclusively originate within the brain. And one of the things that we found was that in terms of this cholesterol flux, that we're talking about, that individuals who had higher levels of those XL HDL seemed to have better biomarkers with respect to Alzheimer's, specifically tau, and some of the markers that are associated with the astrocytes, so gliosis, kind of a thing. And so this is, again, the evidence that we see that to the extent that your brain is able to get that cholesterol debris out from the CNS into the periphery, then this opens up the idea of like, okay, are there things that we can monitor? But again, thinking about it in sort of a systems way, if you have a single point in time, you don't know why, maybe it's higher because there was some export, or maybe it's, higher because for some other reason. And so you need that longitudinal data to, I think, really understand, okay, what's the sort of vector that's going on with your brain in terms of what the neurodegeneration is and how effectively are you clearing it? And I think these are potentially answerable questions, but I think we're a ways off before we can really develop those biomarkers that are going to be informative for this.
Yeah, I mean, it's interesting. You mentioned tau, and you can measure amyloid
in the
and
blood.
tau
Yep.
and NFT and blood, right? Yep. And so I guess the question is, if amyloid in particular and tau to some extent are sort of late markers, how useful are those? Maybe longitudinally. If I think part of the challenge, again, correct me if I've got this wrong or if you think about it differently, but I think part of the challenge is we don't really have enough data on normal aging for those biomarkers to know what is the population reference range outside of the disease.
Correct. Yeah.
Condition.
I
right?
I would agree with that. I think that's what we need is we need to understand these things longitudinally. Right. I think in relationship to, again, having some type of lens, a hypothesis of what we think is going on, and then being able to validate that with these markers, I think is essential.
I mean, at least being able to get some quantitative assessment of neuron death is valuable. But again,
for sure.
I don't think we really know what that looks like at the population level as a function of age or in healthy people. So,
yeah, and it's probably nonlinear too, right?
Yeah, sure.
Yeah,
right.
Yeah.
And you may even have acute phases of a lot of neuron death when you're, I don't know, exposed to some environmental challenge
or a poor night's sleep.
Yeah,
too.
right.
Yeah,
right. We'll probably find out that there's a circadian component to all of this. I'm sure
there
right?
absolutely is. Yeah, in terms of the lipid clearance. Yes.
Yeah. Okay. So diet we talked about. Exercise you touched on. Again, I think you talked about blood flow. Obviously, that's important. Anything else? I mean, again, exercise, one of the probably most potent ways to broadly maintain health, including brain health. Is there anything beyond just exercise good, more blood flow? That's worth saying?
So again, so relating it back to the biology, so there've been a couple studies that have shown that Viagra or Cialis, right, is protective against Alzheimer's. Yeah, exactly. What does that do? That increases cerebral perfusion, right? And so and same thing with like arginine or citrulline, right? Nitric oxide, things that produce that. So if you're increasing that either through exercise or through supplements, then this is, again, the idea is that you are going to potentially be able to better maintain your brain health.
Right. Yeah. And I mean, I was going to talk about this later because I can't go a podcast without talking about rapamycin. But since cerebral blood flow came up, I mean, rapamycin I think is super interesting, obviously, for me because of the context of the biology of aging, but there's also this whole body of literature, even in these terrible mouse models of Alzheimer's disease that we talked about for rapamycin having benefits there. I think one obvious place is in the inflammatory part, right? So in other words, if you have a lot of inflammation in the brain, we know rapamycin is pretty potent anti-inflammatory, including knocking down activated microglia. So that's one place. But there's been a couple of recent studies, I don't know if you've seen them, that I thought were fascinating. They're very small, but they were in ApoE3/4 heterozygotes, looking at both brain volume and cerebral blood flow. And what both of the studies show, this was by MRI and then also, like I said, looking at cerebral blood flow, was improvements in volume in the hippocampus and caudate nucleus and improvements in cerebral blood flow. And that fits with like work from Veronica Galvin in mice on cerebral blood flow with rapamycin. So I thought that was fascinating that it might be hitting this mechanism at a few different places. And there's also, literature. One of the challenges with mTOR and rapamycin is you can find it connected to almost any aspect of biology. But there are connections between lipid metabolism and cholesterol metabolism and mTOR as well. So have you thought at all or looked at rapamycin or mTOR in the context of this model?
I haven't, but it is intriguing. I mean, I do some work on sort of mitochondrial bioenergetics, like so mitochondrial complex 1.
Okay, I want to talk more about that in a minute. So let's put a pin in that, but go ahead.
Yeah. So, and I would be curious because I'm not really familiar with the rapamycin literature, but, to the extent that, AMP kinase is activated and to what extent this sort of shifts, say, substrate utilization.
Okay.
Right.
So let's talk about complex 1. So this is something I've been wondering about for a long time. So this was years ago now in my lab. This was probably like 2009 when we first started thinking about this. We got interested in an observation that in simple animals, when we inhibited mTOR, which is of course what rapamycin does, we could have an outsized effect on lifespan in the context of a variety of mitochondrial defects, right? So we went to Richard Palmer, who was at the University of Washington, who had created this complex 1 deficient mouse. It's called the End-of-Spore knockout mouse. And we asked him if we could do this crazy experiment, which was just give these mice rapamycin. And he's like, well, that'll never work, but sure, here are the mice. So we did it, and it more than doubled their lifespan.
Yeah.
And so, we followed that up a little bit. Other people have, but, we never, I think, really nailed down what the mechanism there was. One of the things we thought about was this substrate utilization. And we definitely saw changes in glycolytic metabolites, lipid metabolism, in the knockout mice treated with rapamycin. But then we also saw decreases in neuroinflammation. But I always wonder, how much does that particular context of mitochondrial dysfunction, complex 1, tie into these neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, and then could some of the effects of mTOR inhibition, rapamycin, cross over, right, by targeting or by alleviating those mitochondrial deficits to have a positive impact on neuronal health or maintenance later on. So I'm interested to know why you think about complex 1 in the context of what I just said. Like, is there a connect point there?
Yeah. So I mean, part of what complex 1 does is, again, NAD and NADH, right?
Right.
So the availability of that, which is then also going to relate to, ATP production.
Right.
So if you're more metabolically flexible, if you have a better adaptive stress response, you can produce more ATP under duress or under these particular circumstances.
Right.
Then you're going to be able to metabolically support your neurons better.
Yeah. So let's double click on this.
Yeah.
So you tell me if I'm remembering this correctly. I'm pretty sure I can pull this out of my head. But so the way this works, right, is glycolysis is an ATP generating process, but most of our ATP comes from the electron transport chain mitochondrial function. Glycolysis is also an NAD to NADH converting process, right? And the way you get that NAD back from NADH is through the electron transport chain as part of the production of ATP.
You know this better than I do.
Okay, so when you block complex 1, you can still make all the ATP you want through glycolysis. The problem is you run out of NAD because it all gets converted to NADH. Is there evidence for that in the context, and maybe this has come out of your model, or maybe you've touched on it in other parts? A lot of people in the longevity space are thinking about NAD precursors and NAD dyshomeostasis. Has that come across your radar as playing an important, potentially causal role in this whole?
For sure, yes. So it has, and so I'm actually on a paper that's looking at End-of-Spore knockout mice as well.
Cool.
Yes. So with Eugenia Trishina at Mayo Clinic.
Yeah.
So yes, we are exploring this. She has some drugs that are, say, partial inhibitors of complex 1. And again, what they are able to do is, and it's sort of counterintuitive, but partial inhibition of complex 1, particularly in, I think it's female mice that are fed a high-fat diet, the inhibition actually, corrects it in terms of a transcriptional profile. So it's beneficial to partially inhibit complex 1. And it seems sort of counterintuitive, but again, I think this has to do with the adaptive stress response. Right. And so when you are able to better sort of shift that and produce more ATP through these sort of alternate routes, then I think you're in better shape.
Okay, that's interesting. Yeah. So one more tangent before we come back to the main thesis. In this End-of-Spore knockout mouse, another really interesting observation was made by Isha Jain and Vamsi Mutha, where they showed that if you put those mice in hypoxia, you can basically completely rescue the disease as long as you keep them in hypoxia. And I mean, their model had to do with oxidative stress, and that in the context of mitochondrial electron transport chain deficiency, you're not using as much oxygen, so you get higher oxygen tension in the brain. That drives oxidative stress. That's a perfectly reasonable model. Tony Grillo, who was a former postdoc of mine, has gone on now independently to look at hypoxia in the context of both mitochondrial dysfunction and Alzheimer's models and, tau hyperphosphorylation, and sees evidence that you can attenuate some of these disease processes with hypoxia. Does that make sense at all in the context of your model in any way that you can connect?
I don't have an answer to that. What I think is fascinating is that, one of the GWAS loci in Alzheimer's is on NFS2,
Okay.
or is associated with
Yeah,
NFS2.
another complex 1
Right.
component.
Yeah. And I think it is sort of counterintuitive because how you sort of manipulate these things, it's not clear why you get some of the outcomes that you
Sure.
do,
Yeah,
right?
yeah.
Like with the hypoxia, you would think that, okay, but another way to sort of also sort of think about it is with hypoxia, it might be similar. You might be simulating exercise.
Right. And there are
Right?
compensatory responses.
Exactly.
HIF1 gets induced.
Yes.
So cause and effect is really, that's why you need a network or a systems model because these are really complicated responses and feedback
Right.
loops.
And so that hypoxia could be, it's probably driving the adaptive stress response with those, which is then sort of enabling some of these other compensatory pathways that are beneficial, which they might not be triggered in some of these other ways, which is why they're beneficial here in this context and may not be beneficial in other ones.
Right. Okay. So back to lifestyle. So we again talked about sleep. Sleep is important for the clearance mechanisms in the brain. Again, anything to say beyond that? I mean, I think we know there's evidence that people who are night shift workers or have fragmented sleep are at higher risk for cognitive dysfunction and Alzheimer's.
Yeah. So with sleep, it is very important, and it's not just each phase of sleep is really sort of setting up the next phase to sort of do its thing, right? And so the key thing, I think, with respect to Alzheimer's and with respect to cholesterol homeostasis and the lipoprotein particles is that, there's a whole, orchestra or orchestration that has to happen to effectively get those lipoprotein particles and that cholesterol out of the brain, right? And so each stage, of non-REM sleep, REM sleep is ultimately going to set you up to be able to get that out of the brain. One of the challenges, I think, too, is that when you have too much debris and say you have ATP deficits, so one of the things that's needed actually for effective sort of sleep and clearing of these lipoprotein particles is, you have to flood the entire system with ATP. And if you don't have that, then you're not going to have very effective sleep. So this is, part of the idea too behind, creatine too.
Yeah, I was going to ask about creatine.
Right?
So you brought it up.
Yeah.
So let's go there.
Yeah. So one of the challenges, though, my understanding is that the creatine currently in its form does not cross the blood-brain barrier effectively. So I think some of the recommendations of really high amounts of creatine are to try and sort of overcome this. Yeah. But I think the rationale is there in the sense that to the extent that it could be doing a few things where if you have, higher, basically ATP buffered capacity, then you're not going to get into this ATP deficit for the neurons, and they're not going to, phosphorylate tau and push it that direction. Yeah. So again, metabolically supporting them would be great. The same thing with the ATP with respect to sleep and being able to facilitate getting rid of those lipoprotein particles.
Right.
Yeah.
So do you take creatine?
I do.
Do you? How much do you take?
I don't take that much. I know I do probably about 5 grams, but I've seen the research that suggests that over 20 is it provides a cognitive benefit.
Right. Yeah. I mean, I think that's what I do too. I mean, I've taken 5 grams a day for a while, and haven't yet bumped up. But I think the data. I mean, it makes sense what you said about more might help you overcome the blood-brain barrier somewhat, and the limited data that's available suggests that up to 20 maybe, or even in some people more, is where you start to see
the cognitive benefit.
Yeah. I don't. Is it your take that there's cognitive benefit or reduced risk or both?
I think it was what I saw. The paper that I remember reading was that there actually was an improved cognitive scores, I think, with the creatine.
Okay. Yeah. We'll have to do a deep dive on that. And this gets at, one of the questions that I sort of already asked, but I'm going to ask again, because I think fundamentally when we're talking about things like creatine or even sleep or diet, right? If we had really useful, accurate biomarkers to know how well is our brain clearing the debris, can we reduce the production of the debris, right?
Yeah.
How do we get there? What do we need?
Well, I think there's I think a few things that we need. I mean, yes, the idea of a biomarker I think is hugely valuable, but the interpretation of the biomarker is just as essential.
Yeah, of course.
Right,
yeah.
And how do you interpret that? Is I think you have to have a context and an understanding of what it's representing.
Yes.
And so I sort of go into this the temporal function of these things, right? So yes, longitudinal is great, but again, being able to understand. And I think this is where what you want is not just a single biomarker, but you want probably a series of biomarkers. So you can measure the coordination of these various processes to understand where somebody is within, say, the development of the disease, or just with normal aging.
Yeah. Okay. So again, thinking about interventions that are interesting or getting talked about a lot. I'm sure you've seen the new paper on lithium.
Yeah.
What are your thoughts there? Does that tie in at all? And
yeah, so there's actually been quite a few papers that have sort of looked at lithium, and I think there's the rationale, as I understand it, is again, so what lithium is potentially going to do is reduce synaptic activity, right? And so again, if you're reducing overall synaptic activity, again, sort of going back to we talked about anxiety, if you're reducing that, then you're conserving energy, right? And to the extent that you're not burning energy, then you're also shifting everything over to being able to better metabolically support the neurons. Now, again, I think this goes to a personalization thing where I think it's an outstanding question to what extent lithium is going to help some people versus others.
Yeah.
And another thing, too, that we had sort of an internal discussion on this, but lithium levels vary substantially across the country.
Absolutely.
Yeah. And so to what extent, depending on where you live and how much you have already relative to supplementation, I think is also going to
And I mean
determine
that's one of the reasons why, that's one of the lines of evidence that makes that model compelling to me, is that if you look at the natural variation in lithium levels across populations, you do see that it correlates at least with Alzheimer's disease incidence rates, right?
Yeah.
I guess the other thing that I thought was interesting about the recent Nature paper was the connection to amyloid beta, in the sense that the plaques might be titrating lithium out of tissues, which would sort of lead to this vicious, even if it's a later event, right? It may be supporting this vicious cycle, right?
Yeah,
so
I agree with that.
The biochemical part to me is what really makes the newer data, in addition to what exists, sort of crosses that threshold for me. We're like, okay, that makes a ton of sense.
Yeah. So there was a paper that I was on, I think it was released last year or 2 years ago. It was with Todd Goldie, who's at Emory. And he really looked at the contents of amyloid plaques. And we call them amyloid plaques, but they have over 20 different proteins in them.
Sure.
Yeah. And so one of the things too, and I'm not sure that this made it into the paper, but they seem to also absorb some of the growth factors that help to sort of sustain neurons as well. And so your idea too of the lithium sort of being absorbed into there, I think would be
maybe
consistent
other
with something
things
like
as well.
Yeah, exactly.
Yeah, cool. Yeah. Okay, so any interventions we haven't talked about that you're excited about or you're thinking about based on your own research and how it might tie in?
I think we've touched on most of them. Again, it is it's a complex problem. So I don't know. None of these are magic bullets.
Yeah. I agree with that for sure. Yeah.
And I think the real challenge is figuring out who is going to benefit from what.
Right. The personalization.
The personalization.
Piece. Yeah. So one thing we haven't touched on yet, talked about diet, but in the context of, people hear a lot about the microbiome and its role in Alzheimer's disease. How does that play into this?
Great question. And I think that there is some really interesting data out there where, I think they've done sort of some, microbiome transplants in some of these, 5X FAD, some of these, early onset amyloid mouse models, and they do see a benefit. And I think there's some hypotheses around what that might be doing or how that might be impacting things. It's less clear to me though on that. I mean, one of the things that I do think is interesting with the microbiome is that so downstream of cholesterol metabolism are bile acids, right? And so you have primary and secondary bile acids, and the bile acids, secondary bile acids are all produced by the gut microbiome. And to what extent there might be a feedback loop between those bile acids and cholesterol homeostasis, I think is an interesting thing that I would like to explore.
And do we know anything at this point about drugs that modify bile acid?
Not really. I haven't really seen anything. There could be stuff out there, but I've not seen anything specific on that. Yeah.
So given, everything that you've uncovered in your research, what can somebody today in their 30s or 40s do to really meaningfully reduce their risk of developing Alzheimer's disease?
Yeah, so I think we've touched upon those, and they are synonymous with what is healthy living, right?
Yeah.
I don't think there's any difference. I mean, your body health and your brain health, I think, are closely connected in many ways.
Yeah.
So I think understanding those, I do think sleep is just crucial. So especially as a younger person, that's when things sort of get set in motion. And to the extent that you are subjecting yourself to sleep deprivation, high stress kind of a thing, it definitely has a cumulative toll.
What about the role of human relationships, right? So there's been accumulating body of work, right, on social isolation as a risk factor for dementia. So how does that play into this?
Yeah, so I think this is also part of how you are wired in terms of your neuronal activity, and to the extent like extroverts also live longer, right? And so I think the idea here is that when through brain development and through interaction, there's sort of the use it or lose it kind of phenomena with synapses and neurons, and to the extent that you're continuing to sort of maintain that muscle, then I think you're also going to be shifting things over to having more neurons that are active and useful than not.
So you think it's at least part almost an environmental enrichment?
Yeah, I
effect?
do. Yeah, I do. I think, and it could be multiple, have multi-factors to that too, right? In terms of reducing anxiety, right? So
Sure.
if you're more connected, then you
Right.
tend to have less anxiety,
Right.
As well as the sort of stimulation.
Right.
Yeah.
Okay. If you could flash forward 20 years from now, what do you hope people will say about your work?
Yeah, so 20 years from now, what I hope is that in terms of the model that we're working on now and its application, I really believe that we will have solved the problem of Alzheimer's for many, if not most people. In that, I think we will have effective biomarkers. I think we will have effective interventions. I think we will be able to understand how all these risk factors contribute to the disease. And I think people are going to have to be informed in terms of like, okay, if they want to have a night out partying, okay, what is that going to look like?
Cost?
What's the expense?
Yeah.
And I think we might have an idea and be able to quantitate that to some extent.
So do you think we'll, and I'm asking you to speculate, and the people always ask me to speculate on where longevity is going to be in 20 years, and I hate it half the time. But nonetheless, I'm going to ask you to do it. Do you think we will get to the point where we have a cure for Alzheimer's disease, or we'll know enough where we can slow the progress sufficiently that it's not going to impact most people?
Yeah, so I like the distinction in the sense that there can be things to prevent it, but then if somebody develops or has Alzheimer's, will we be able to reverse it? And I am hopeful that we will ultimately be able to do that. Again, going back to this hypothesis that we have around cholesterol homeostasis, if we're able to correct that, if we're able to identify those things that are contributing to that metabolic deficit that are causing the neuronal death, if you've stopped that, then to what extent you can regenerate neurons, I think that there is evidence that you can do that. There's probably things that we can do to even boost that even more so. So I'm hopeful within 20 years, given 20 years, especially the accelerating rate of progress, I think we can ultimately do that.
Great. Let's see. What's one belief about Alzheimer's disease you wish you could erase from the public's mind forever?
Yeah, so I think there's a lot of sort of thinking that it is a death sentence. That particularly if you're an E4 carrier, that there's nothing you can do. And I think there's plenty that you can do. I think you can be proactive, and I think it does make a difference. I think we have very strong evidence that it does make a difference. And so, again, what do you care about? Do you quality of life, longevity? You want to have a good life, and what does that look like? I think that's personal determination. But I think you are empowered, and I think we're becoming even more empowered to make those decisions.
Yeah, I think that's a super important statement, because you're right. A lot of people have the perception that if they, like you said, if they're ApoE4 homozygotes, or if they are experiencing the early stages of cognitive decline, that it's an inevitable path from here, and that couldn't be more untrue. I wouldn't want to say that you can completely offset all of the risks associated with.
Not yet,
but it's getting pretty close. Like I think you can go most of the way to offsetting the risk if you're an ApoE4 homozygote, if you really, really dial in the factors that you can control around lifestyle and early intervention with medications.
Yeah, you definitely, I think, can have an impact on that. Yeah. I think this is particularly relevant for people who have a family history. They have seen their parents or loved ones go through this. And I think that in particular, if we sort of I think, incorrectly ascribe like, well, this is a genetic thing, that this is not something I can necessarily escape. And I think that's even more prominent in people who see this firsthand. And it's those individuals that I think, ultimately they are the worried well, or they're a subset of the worried well. And I do think that there's hope for them.
Right. Great. On a personal level, how has your research changed your own approach to health?
So I am fairly health conscious.
Were you before you started doing this research?
Yeah, I kind of. I mean, I've always enjoyed exercising.
Yeah.
I'm a cyclist, so I like to bike. I'm pretty active. I like to hike. Again, it goes to sort of quality of life.
Do you pay attention to your sleep more now?
I do prioritize it. I prioritize my sleep, and I think that's not just for a longevity thing, but I think, again, going to quality of life. I don't think that there's little that can have as much of an impact on your quality of life as sleep. Yeah. In my opinion, and in my experience as well. Yeah.
Okay. Anything we haven't touched on that you feel like it's important to get out there?
I think we've covered a lot of bases. Yeah.
Awesome.
Yeah.
Well, thank you so much. This has been a lot of fun.
You bet. My pleasure. It's been a lot of fun talking about this.